In this lecture, we're going to focus on what's called the omics, and we're going to concentrate a little bit more on proteomics, pharmacogenomics, and bioinformatics. At the end of the lecture, you should be able to define the following terms: transcriptomics, metabolomics, nutrigenomics, pharmacogenomics, pharmacogenetics, biomarkers, SNPs, bioinformatics, and computational biology. You should be able to identify at least 3 uses for proteomics and molecular diagnostics, list some of the methods used to identify and separate proteins, identify several biomarkers of disease that can be used for diagnosis, identify at least 2 uses for pharmacogenomics in healthcare, describe at least one example of pharmacogenomics such as HER2, Herceptin, Cytochrome P450, or TPMT, list the benefits of pharmacogenomics, list the barriers of pharmacogenomics, and list the areas where bioinformatics is currently in use, as well as one example such as sequence analysis. The long-term omics goals. Well, omics have the potential to deliver biomarkers for health and comfort, reveal early indicators for disease, assist in differentiating responders from non-responders, and discover beneficial and harmful components for specific diseases. Now, all of these omics are a relatively new field. So now that many genomes have been sequenced, including the human genome, their transcriptomes can be predicted with better accuracy. And genome-wide expression analysis can deliver a comprehensive view on all genes active at a given time in a sample. So the fact that the Human Genome Project was completed and so many organisms have been sequenced, we can now take all of this sequence information and utilize it in all of the various omics fields. So here are some of the omics. Transcriptomics is the study of the transcriptome, which is the complete set of RNA transcripts produced by the genome at one time. Metabolomics is a systemic study of the unique chemical fingerprints that specific cellular processes leave behind. So the metabolome represents the collection of all metabolites in a biological organism, which are the end product of its gene expression. So while messenger RNA gene expression data and proteomic analyses don't tell the whole story of what might be happening, the metabolic profiling can give an instantaneous snapshot of the physiology of that cell. Nutrigenetics is identifying how genetic disposition affects susceptibility to a diet. Genetic disposition is measured in single nucleotide polymorphism, copy number polymorphisms, and epigenetic phenomena. Nutrigenomics addresses how diet influences gene transcription, protein expression, and metabolism. The challenge is integrating genomics or gene analysis, transcriptomics, gene expression analysis, proteomics, global protein analysis, and metabonomics or metabolite profiling to define a healthy phenotype. The long-term goal is personalized nutrition for maintenance of individual health and prevention of disease. The proteomic concepts. So the human genome only comprises 25,000 genes. That's much less than what was originally estimated that the human genome would contain. The human proteome is estimated to encompass several 100,000 proteins. So the overall dynamic range of individual protein levels spans 6 orders of magnitude in a cell, and at least 10 in the human body. And protein expression changes over time in certain situations. So a proteomic study must rely on representative snapshots of protein patterns. Proteomics is the large-scale study of proteins, particularly their structures and functions. Proteins are vital in living organisms. They're the main components of the physiological pathways of the cell. So the term proteomics was coined to make an analogy with genomics, the study of genes. The proteome of an organism is the set of proteins produced by the organism during its life, and its genome is the set of genes. The proteome of a cell under a particular stimulation or environmental situation is the set of proteins in it. So the word proteome derives from proteins and genome, since the genome is what encodes those proteins. Proteomics is often considered the next step in the study of biological systems after genomics. It's more complicated in genomics because while an organism's genome is constant, a proteome differs from cell to cell and constantly changes through the biochemical interactions with the genome and the environment. So one organism has radically different protein expression in different parts of its body in different stages of its life cycle, as well as in different environmental conditions. Another major difficulty is the complexity of proteins relative to nucleic acids. Proteomics gives a much better understanding of an organism than genomics. So the level of transcription of a gene gives only a rough estimate of its level of expression into a protein. So a messenger RNA produced in abundance may be degraded rapidly or translated inefficiently, resulting in a small amount of protein. So you really can't tell how much protein will be made based on the level of transcription that you might see. Many proteins experience post-translational modifications that also affect their activities. So this would be phosphoproteomics and glycoproteomics would be used to study these post-translational modifications. Also, many transcripts give rise to more than one protein through alternative splicing or alternative post-translational modifications. And many proteins form complexes with other proteins or with RNA molecules and only function in the presence of these other molecules. Current research in proteomics requires first that the proteins are resolved, and protein separation can be performed using two-dimensional gel electrophoresis, where you spot your protein and you visualize it using a variety of chemical stains or fluorescent markers. So when you run your protein in a gel, you can quantify the protein by the intensity of the stain. Once those proteins are separated and quantified, they're then identified. So those individual protein spots are cut out of the gel and cleaved into peptides with proteolytic enzymes. The peptides are then identified by mass spectrometry. So usually MALDI-TO, or matrix-assisted laser desorption ionization time of flight mass spec, is used. And in this procedure, a peptide is placed on a matrix, which causes the peptide to form crystals. The peptide on the matrix is ionized with a laser beam, and an increase in voltage at the matrix is used to shoot ions towards a detector, in which the time it takes an ion to reach the detector depends on its mass. So the higher the mass, the longer the time of flight of the ion. So in a MALDI-TO mass spec, the ions can also be deflected with an electrostatic reflector that also focuses the ion beam. So the masses of the ions reaching the second detector can be determined with very high precision, and these masses can reveal the exact chemical composition of the peptides and their identities. Protein mixtures can also be analyzed without prior separation. So this procedure begins with proteolytic digestion of the proteins in a complex mixture. The resulting peptides are often injected into a high-pressure liquid chromatography column, or HPLC, and this is going to separate peptides based on hydrophobicity. HPLC can be coupled directly to the time of flight mass spec using electrospray ionization. Peptides eluting from the column can be identified by tandem mass spec. The first stage of tandem mass spectrometry isolates individual peptide ions, and the second breaks the peptides into fragments and uses the fragmentation pattern to determine the amino acid sequence. Labeling with isotope tags can then be used to quantitatively compare protein concentration among two or more protein samples. Cancer proteomics is enabling mapping of the patterns of proteins involved when normal cellular pathways are hijacked in support of malignant growth. In cancerous tissue, some of the proteins critical for normal communication are damaged, are inactive, are overactive, or are completely missing. The full set of deranged and dominating proteins at work disrupting cellular communications can vary from one cancer type to another cancer type, and they may also vary from one patient to another with the same cancer type. So another major form of molecular diagnostics looks beyond genes to an investigation of the proteome. So the term proteome refers to all the proteins produced by a cell, and the study of these proteins is proteomics or proteomics. Cancer proteomics is enabling mapping of the patterns of proteins involved when these normal cellular pathways are hijacked in support of malignant growth. So cancer researchers are devising clever ways to capture data on each cancer's characteristics and their unruly set of proteins. So biopsy or blood samples from cancer patients are the starting point, and various methods are used to extract and analyze the sets of interactive proteins. So for many proteins in the cell, the addition of a small molecule, which is a phosphate, acts as a switch that activates that protein, and this is called phosphorylation. To preserve a cell's phosphorylation state and capture an accurate pattern of proteins interacting in a cancer cell, researchers handle the biopsy or blood specimens very carefully. So they have to see that samples are quickly treated with enzymes to block the removal of phosphates from proteins. And this is going to enable the identification of a protein pattern almost identical to what was in the cell at the time of collection. So this is going to enable researchers to really look at these protein patterns within cancerous cells and compare those to normal cells and try to really look at those differences. Understanding the proteome, which is the structure and function of each protein, and the complexities of protein-protein interactions is critical for developing the most effective diagnostic techniques and disease treatments in the future. So an interesting use of proteomics is using specific protein markers to diagnose disease, and a number of techniques allow to test for proteins produced during a particular disease, which helps to diagnose the disease quickly. And some of these techniques are Western blots, immunohistochemical staining, enzyme-linked immunosorbent assays or ELISAs, and mass spec. So biomarkers for diagnostics. In Alzheimer's disease, elevations in beta secretase creates an amyloid beta protein, which causes plaque to build up in the patient's brain, which is what is one of the causes of dementia. So targeting this enzyme decreases the amyloid beta protein and slows the progression of disease. So a procedure to test for the increase in amyloid beta protein is immunohistochemical staining, where antibodies bind to specific antigens or biological tissue of amyloid beta protein. And another disease, heart disease, this is commonly assessed using several key protein-based biomarkers. So standard protein biomarkers for heart disease include interleukin-6, interleukin-8, serum amyloid A protein, fibrinogen, and troponins. So the CTN1 cardiac troponin 1 increases in concentration within 3 to 12 hours of initial cardiac injury and can be found elevated days after an acute myocardial infarction or a heart attack. So a number of commercial antibody-based assays as well as other methods are used as primary tests for acute myocardial infarction. Proteomic analysis of kidney cells and cancerous kidney cells is producing promising leads for biomarkers for renal cell carcinoma and developing assays to test for renal cell carcinoma. So in kidney-related diseases, urine is a potential source for biomarkers, and it's been shown that the identification of urinary polypeptides as biomarkers of kidney-related diseases allows diagnosis of the severity of the disease several months before the appearance of the pathology. Various technologies are used in proteomic studies. So these technologies are both 1 and 2 dimensional gel electrophoresis, X-ray crystallography, nuclear magnetic resonance, tandem mass spec, reverse phase chromatography, MALDI-TO spec, affinity chromatography, yeast to hybrid techniques, FRET or fluorescence resonance energy transfer, X-ray tomography, and software-based image analyses. So now we're going to move into pharmacogenomics. Pharmacogenomics is the study of how an individual's genetic inheritance affects the body's response to drugs. So the term comes from the words pharmacology and genomics and is the intersection of pharmaceuticals and genetics. Pharmacogenomics holds the promise that drugs might one day be tailor-made for individuals and adapted to each person's own genetic makeup. Environment, diet, age, lifestyle, and a state of health can all influence a person's response to medicines, but understanding an individual's genetic makeup is thought to be the key to creating personalized drugs that have greater efficacy and safety. Pharmacogenomics combines traditional pharmaceutical sciences such as biochemistry with annotated knowledge of genes, proteins, and single nucleotide polymorphisms. Pharmacogenomics refers to the general study of all of the many different genes that determine drug behavior. Pharmacogenetics refers to the study of inherited differences or variations in drug metabolism and response. So the distinction between the two terms is considered arbitrary, and the two terms are now often interchangeable. Currently, physicians prescribe medication through a trial and error method of matching patients with the right drugs. So if the prescribed medicine doesn't work for the patient the first time, the physician will try a different drug or dosage and repeat this process until the patient improves. So as pharmacogenomics becomes more advanced, physicians may eventually be able to prescribe medication based on the individual patient's genotype, which will maximize the effectiveness and minimize side effects. So microarrays, we discussed microarrays several times previously. Microarrays are also an important technique used in pharmacogenomics, which, as we know, is an area of research that studies why certain drugs work in combination with particular genetic expression patterns, but not with others. So this information is being used to design new drugs that target altered cells without affecting normal cells. So microarrays are being used, so if you look at this slide, we have patient A and patient B. They have the exact same cancer. You could then use microarrays to look at the gene expression profile of each individual's tumor. Then you can determine if the drugs block the active site. So you could see, although these two patients have the exact same cancer, in patient A, the drug does block the active site, but it doesn't block the active site in patient B. And that's because the gene expression profile of their tumors is different. So the HER2/neu protein, Herceptin, is a very good example of pharmacogenomics. In the 1980s, researchers discovered that some women who had a very fast-growing breast cancer expressed extra copies of a gene called HER2/neu. The genes were producing many copies of a protein that appeared to be driving the growth of the cancer cells, so enhancing their growth. So you can see in this slide that there's a breast cancer patient, and this patient's cells has the HER2/neu gene, and the cells have the HER2/neu receptor, which are binding to a growth factor, causing these cells to grow very rapidly. So in the early 1990s, an antibody was developed that latches onto the HER2/neu proteins on the surface of a cancer cell, and it stops the proteins from spurring on the cancer cell growth. And through this stop signal, it can also stop the propagation of survival signals within the cancer cell. So some women were given this experimental antibody, and they saw that their cancer growth slowed down or stopped altogether when the Herceptin treatment was combined with cytotoxic chemotherapy. So Herceptin was approved by the FDA in late 1998 as a therapy for women who test positive for high levels of the HER2/neu protein. And this is about 25 to 30% of all breast cancer patients. So you can see, again, you have a breast cancer patient, you have the HER2/neu receptors on the cell surface. The growth factor is there. However, the drug Herceptin blocks the receptor so that the growth factor cannot bind to the growth receptor. So that tumor then is slowed down its growth or possibly even stops growing. So this new technology allows researchers to study patient populations, collecting expression patterns of tumors before any treatment is given, and then analyzing treatment outcomes, by doing this, scientists hope to identify expression patterns that can accurately predict a good response to treatment. So in the future, before any treatment is given, doctors may be able to predict a patient's response to chemotherapy or radiation based on this research, which could eliminate a lot of that clinical guesswork. So by looking at expression patterns found in a single blood sample or a small tissue biopsy sample, doctors may be able to tell which of the available drugs or radiation treatment is going to work best for that individual patient and which drugs are not going to work. So clinicians will be better able to plan effective therapies at the proper doses for individual cancers. So here we have with the advent of microarrays, pharmacogenomic research has entered a new level of sophistication. So the technology allows researchers to study patient populations collecting expression patterns of tumors before any treatment is given. So you have patient A and patient B. The microarray is going to give you a gene expression profile, and you can then, even though these patients might have the same exact cancer, the expression profile differs, so the effective treatment may differ. So you can actually give each patient a different treatment that's going to be effective for their cancer, even though technically they're diagnosed with the same cancer. So SNPs are single nucleotide polymorphisms. For pharmacogenomics to be effective, markers must be found that indicate the connection between drug response and genetic makeup. So the markers that companies are pursuing are known as single nucleotide polymorphisms, or the SNPs. So SNPs are defined only in relation to a population and are variations in DNA at a single base that are found in at least 1% of the population. So drugs are broken down in the body by proteins, and not all of these proteins are identical in each individual. So SNPs can result in subtle differences in proteins that translate to major differences in how the protein functions. So for example, as many as 40% of individuals have a SNP that causes a deficiency in a protein called Cytochrome P450. And this is the CYP2C9, which is important in drug metabolism. So deficiencies in this protein can impair the metabolism of as many as 1 in 5 medications currently on the market. So the cytochrome P450 family of liver enzymes is responsible for breaking down more than 30 different classes of drugs. DNA variations in genes that code for these enzymes can influence their ability to metabolize certain drugs. So less active or inactive forms of the CYP enzymes are unable to break down and efficiently eliminate drugs from the body, and this can cause overdose in patients. So clinical trials use genetic tests for variations in cytochrome P450 genes to screen and monitor patients. Many pharmaceutical companies screen their chemical compounds to see how well they're broken down by variant forms of the cytochrome P450 enzymes. Another enzyme called TPMT, or thiopurine methyltransferase, plays an important role in the chemotherapy treatment of a common childhood leukemia by breaking down a class of therapeutic compounds called thiopurines. So a small percentage of Caucasians have genetic variants that prevent them from producing an active form of this protein. So as a result, thiopurines elevate to toxic levels in these patients because the inactive form of the enzyme is unable to break down the drug. So now doctors can use a genetic test to screen patients for this deficiency, and then the TMPT activity is monitored to determine appropriate thiopurine dosage levels. So the benefits of pharmacogenomics: you get more powerful medicines. Pharmaceutical companies will be able to create drugs based on the proteins, enzymes, and RNA molecules associated with genes and disease. It will facilitate drug discovery and allow drug markers to produce a therapy more targeted to specific diseases. And this accuracy is going to maximize therapeutic effects and also decrease damage to healthy cells. Also, we'll get better, safer drugs the first time. So instead of that trial and error method of matching patients with the right drugs, we'll be able to analyze a patient's genetic profile and prescribe the best drug right at the start. So this takes the guesswork out of finding the right drug. It'll improve recovery time. It'll enhance safety. It's going to lessen the adverse reaction that a person might have. So pharmacogenomics has the potential to dramatically reduce the estimated 100,000 deaths and 2 million hospitalizations that occur each year in the U.S. as a result of adverse drug response. Pharmacogenomics is also going to provide more accurate methods of determining appropriate drug dosages. So common methods of basing dosage on weight and age will be replaced with dosages based on the person's genetics. So how well does that person's body and cells process the medicine and metabolize it? So this is going to maximize the therapy's value and decrease the likelihood of overdose. Some of the other pharmacogenomic benefits, including advanced screening for disease. If a person knows their genetic profile, they can make adequate lifestyle and environmental changes at an earlier age to lessen the severity of the genetic disease. It'll provide better vaccines made of DNA and RNA and genetic materials. It'll improve the drug discovery and approval process, and it'll decrease the overall cost of health care. So the future potential is, if you think about going to the doctor's office and instead of getting prescribed a medicine based on what's currently available for that disorder, you can have a genetic test done that indicates which medication is right for you. So this test might predict how you would respond to a drug or determine whether you were in a group of patients who would suffer severe side effects. The barriers to pharmacogenomics include the complexity of finding gene variations that affect drug response. So SNPs, those single nucleotide polymorphisms, occur every 100 to 300 bases. So millions of SNPs must be identified and analyzed to determine if they are involved in a drug response. So another limitation is knowing which genes are involved with each drug response. So many genes are likely to influence responses. So finding out exactly which ones is time-consuming and very complicated. Another barrier is limited drug alternatives, so only 1 or 2 approved drugs may be available for the treatment of a particular condition. So if a patient has a gene variation that those drugs would cause them to have a detrimental response or an overdose, there may not be any other alternatives for treatment. There's also disincentives for drug companies to make multiple pharmacogenomic products. So pharmaceutical companies have been successful with their one-size-fits-all approach. It costs hundreds of millions of dollars to bring a drug to market. So will pharmaceutical companies be willing to develop numerous types of drugs that may only serve a very small portion of the population? Also, educating healthcare providers is a barrier. So introducing multiple pharmacogenomic products to treat the same condition for different population subsets is going to complicate the process of prescribing and dispensing drugs. So future medicine, we already know that if we sample tumor tissues from 100 different people, these tissues would have a molecular makeup that would break up into different categories. So essentially, each patient has a different disease, but in the case of, let's say, breast cancer, we just happen to be saying that all 100 women have breast cancer. So once we get people with the right disease diagnosis, the disease definition will change from you have breast cancer to you have a cancer in the breast with the molecular profile A, B, C, or D. And the treatments of these diseases are going to be different. And that leads us into bioinformatics. So the terms bioinformatics and computational biology are often used interchangeably. Bioinformatics refers to the creation and advancement of algorithms, computational and statistical techniques, and theory to solve formal and practical problems inspired from the management and analysis of biological data. Computational biology refers to hypothesis-driven investigation of a specific biological problem using computers, carried out with experimental or simulated data with the primary goal of discovery and advancement of biological knowledge. So put more simply, bioinformatics is concerned with the information, while computational biology is concerned with the hypotheses. So some of the areas of bioinformatics include sequence analysis, genome annotation, computational evolutionary biology, analysis of gene expression, protein expression, analyzing mutations in cancer, prediction of protein structure, comparative genomics, modeling biological systems, and high-throughput image analysis. So with sequence analysis, there is a growing amount of data. Every single possible organism has been or is in the process of being sequenced. So it has become impractical to analyze all the DNA sequences manually. So what we use are computer programs to search the genome of thousands of organisms containing billions of nucleotides. So these programs compensate for mutations in the DNA sequence in order to identify sequences that are related but not identical. Another aspect of bioinformatics in sequence analysis is the automatic search for genes and regulatory sequences within a genome. So not all of the nucleotides within a genome are genes, as we know. You have your exons and your introns, or your intergenic regions. So within the genome of higher organisms, large parts of the DNA don't serve an obvious purpose, or we don't know yet what these regions do. So right now these are referred to as junk DNA. So these may contain unrecognized functional elements, and we actually know that some of these intergenic regions do contain regulators and things such as regulatory small RNAs that do have a function. So bioinformatics helps to bridge the gap between genome and proteome projects. So for example, the use of DNA sequences for protein identification. And the assembly of the human genome is one of the greatest achievements of bioinformatics. So omic data success, all of these different omics, your nutrigenomics and metabolomics and transcriptomics and proteomics, the success depends on the resolution of analytical challenges. The maturity of the bioinformatic tools and the quality of the databases vary. So you need to constantly be improving the tools in order to analyze all of this data. Fundamental research is needed to understand the interplay between individual disposition, health, environment, as well as diet, and being able to answer which health issues can be addressed by certain means. So for nutrigenomics, a nutritional mean. For proteomics, which protein is involved in a specific health issue. Which health or disease condition manifests at which omic level, and which omic platform or in vitro and in vivo model and biological sample will be best. The benefits of the omics is that they deliver biomarkers for individual disposition and health maintenance. The biomarkers at the transcript, protein, and metabolite level will develop into indicative and predictive sets of molecules, and the diagnostic power can't be uncoupled from predetermined genetic disposition. So the major omics challenges is integration of gene and protein expression profiles with metabolic fingerprints in where you have new findings all the time, and more understanding is needed regarding how to select relevant subsets of information, as well as the merging of this information. Health and wellness are generally poorly understood, and we know that the environment, genes, and health are all interconnected. So this makes the omics challenging because you don't just have genes involved, you have all these other things. So one of the challenges of systems biology and functional genomics is to integrate the proteomic, transcriptomic, metabolomic, and all the different omic information to give a more complete picture of living organisms.